So imagine taking a single 3D picture of your brain today, like right now, while you feel perfectly healthy.
Right, completely asymptomatic.
Exactly. And then having an artificial intelligence generate this flawless decade-long video showing exactly how your specific brain is going to shrink and, fold and age.
It's wild. You're basically watching an inevitable disease progress years before it ever happens in the real world.
Yeah. And if you are a student diving into medical AI or longitudinal neuroimaging, understanding how to build that kind of predictive model is, well, it's essentially the holy grail of the entire field.
Oh, absolutely. Because the clinical implications of mapping out that future geometry, they are staggering.
Right.
Neurodegenerative conditions, particularly Alzheimer's disease, they don't just happen overnight. They are characterized by this gradual spatially heterogeneous atrophy.
Spatially heterogeneous, meaning?
Meaning that different structures inside the skull are shrinking at totally different rates.
Ah, okay.
Yeah, and this structural decay begins years, often decades, before a patient, forgets a name or shows any clinical symptom of memory loss.
Wow, decades.
Exactly.
Well, today we are exploring the bleeding edge of how we actually generate that predictive map. So welcome to the deep dive, everyone.
Glad to be here.
Our focus today is a recent highly technical research paper. It's titled Generative Modeling of Neurodegenerative Brain Anatomy with 4D Longitudinal Diffusion Model.
A bit of a mouthful.
Yeah, quite the title. But it was put together by researchers from the University of Virginia and Weill Cornell Medical College. And our mission today is to figure out how they managed to predict the future of a human brain from just one baseline scan.
Which is a massive leap. But to appreciate that leap, we first have to ground ourselves in the reality of medical research.
Right.
Like, before we even touch the artificial intelligence, we have to talk about the physical data.
The missing time problem, right?
Exactly. If you are studying how a brain changes over a decade, you need longitudinal data. You need a continuous timeline of MRI scans from the exact same individual.
But that's hard to get.
Oh, in the real world of neuroimaging, that data is notoriously sparse.
See, I always trip up on this when we talk about healthcare data because we constantly hear about the era of, big data.
It's right, the buzzword.
Yeah, where hospitals are collecting millions of data points every single day. So why is it so incredibly hard to get a continuous timeline of one person's brain decay?
Well, so big data in healthcare usually implies passively collected data, like heart rate monitors, blood tests, standard charting.
Stuff that just happens in the background.
Exactly.
Yeah.
But longitudinal neuroimaging requires active, grueling participation. I mean, consider the patient population here.
Right. Usually elderly.
Elderly individuals, yeah. And many of whom are actively experiencing the terrifying early stages of cognitive decline.
Oh, man.
And you're asking them to travel to a research facility, lie perfectly still inside a noisy, claustrophobic MRI tube for an hour.
Which is stressful enough on a good day.
And you need them to do this every 6 months for 10 years.
Okay, yeah. When you frame it like that, the logistical nightmare becomes pretty obvious.
The dropout rate is just incredibly high.
Mm-hmm.
Patients move away, or, they get too sick to travel.
Or they lose their transportation.
Exactly. Or they simply pass away. Even the most dedicated participants might just miss an appointment.
Right.
So a researcher pulls up a patient's file and finds like a high-resolution scan at year 1, maybe another scan at year 2, and then absolute silence until year 5.
It makes me think of trying to follow the plot of a movie by only looking at 3 random screenshots.
That's a great way to put it.
Or like a flip book where someone ripped out 90% of the pages.
Yes, the ripped-up flip book.
If we just have that brain scan from year 1 and a follow-up from year 4, we are entirely blind to what the disease was doing during years 2 and 3.
And that sparsity is the major roadblock in clinical neuroscience today. It severely restricts our ability to model the continuous anatomical changes of a disease.
Which leaves us with a pretty massive question.
A multi-million-dollar clinical question. Can we computationally simulate an individual's future disease progression from just that one initial baseline scan and seamlessly fill in those missing pages?
Right. And given the recent explosion in generative AI, I feel like the immediate assumption a lot of students might make is, well, just use existing technology.
Sure, just use an image generator.
Yeah. We see AI generate hyper-realistic videos of entirely fictional people every single day. If we're just missing images of a brain, why not feed the year 1 and year 4 scans into a standard image generator and just ask it to fill in the blanks?
Well, researchers have aggressively pursued that exact path. The paper actually outlines the evolution of generative modeling in medical imaging.
Okay.
And it started with GANs, or generative adversarial networks.
Right, GANs. That is the architecture where 2 neural networks basically fight each other, right?
Exactly.
Like, one acts as the forger trying to paint a fake brain scan, and the other acts as the detective trying to spot the fake.
The adversarial process, yeah.
And over time, the forger gets incredibly good at making realistic images.
It is powerful. But in the context of neurodegeneration, it runs into a brick wall called mode collapse.
Okay, mode collapse. How does that manifest when we're talking about medical imaging?
So mode collapse means the AI finds one specific output that consistently fools the detective network. And it just gets lazy.
Lazy.
Yeah, instead of learning the vast, highly individualized variability of how Alzheimer's affects different human beings, the network just starts generating the exact same generic sick brain over and over again.
Wow.
It completely loses the individual patient's unique anatomy.
So the GAN essentially just gives us a biologically useless average.
Exactly, which is why the field largely moved on to denoising diffusion probabilistic models, or DDPMs.
DDPMs, and these are the engines behind the massive text-to-image generators we see dominating the tech world right now.
Right. Yes. And diffusion models are mathematically brilliant. During training, they take a clean, high-resolution image and they gradually add Gaussian noise to it.
Like static on a TV.
Exactly, step by step, until the image is nothing but unrecognizable static. Then, the AI learns to reverse the process. It learns how to remove the noise to reconstruct the high-resolution image.
Okay, but if they are that powerful, why can't a diffusion model just look at a healthy brain and denoise it into a future diseased brain?
The crucial distinction, and this is the fatal flaw the researchers highlight in the paper, is what these diffusion models are actually manipulating.
What do you mean?
Standard DDPMs manipulate image intensities. They are adjusting the brightness and color values of pixels on a 2D grid.
Okay.
They have absolutely no concept of the underlying biological geometry.
Oh, so it's basically acting like a highly advanced Photoshop smudge tool?
That's exactly it.
It knows that an older brain usually has darker pixels in a certain area, so it smudges the pixels to match. But it doesn't actually know it's looking at a physical, three-dimensional biological organ.
It doesn't understand physics, it doesn't understand tissue boundaries, and it certainly doesn't understand that a brain is a solid, interconnected mass of tissue that morphs over time.
It's just guessing colors.
Right. You're just predicting what shade of gray a specific square should be based on statistical probability.
And I imagine the consequences of that ignorance are pretty wild.
Oh, they are.
Because when these pixel-based models try to generate the progression of Alzheimer's, they produce anatomically implausible samples.
Because the AI doesn't understand physical reality, you get severe structural distortions. The text mentions brain tissue folding in on itself, discontinuous warping.
And my personal favorite consequence, tearing.
Yes, tearing.
The AI will generate a sequence where the brain tissue literally rips apart like wet paper.
Which is biologically impossible. An atrophied brain doesn't tear, it undergoes a structural geometric breakdown.
Right.
Inside your brain, you have these fluid-filled spaces called ventricles. As the surrounding gray and white matter dies and shrinks, those ventricles expand to fill the empty void.
So it's a fluid shift.
It is a continuous, interconnected deformation. If an AI generates a scan where the temporal lobe is physically severed from the rest of the brain because of pixel hallucination.
Like a glitch in the matrix.
Yeah. That image cannot be used for clinical research. It is medically useless.
So we need an AI that fundamentally respects the laws of physics and biology.
Which brings us to the core innovation of this research paper.
To solve the tearing brain problem.
Right. To solve that, this team abandoned the 2D pixel smoothing mindset completely. They built a 4D geometric framework.
Okay, this is where the paradigm shifts. They propose a novel 4D diffusion-based generative framework.
Instead of operating in the space of individual image pixels, it operates in the space of topology-preserving spatiotemporal deformations.
Okay, topology-preserving spatiotemporal deformations. Let's break that down for any student trying to conceptualize this.
Sure.
So 4D simply means we are dealing with 3D space, the complete volumetric shape of the brain, plus time, which is the fourth dimension. But instead of drawing a brand new brain from scratch for year 2, year 3, and year 4, the AI generates what they call a velocity field.
Yes, the velocity field is the secret engine of this entire process.
The best way I can think of it is like mapping out ocean currents.
Okay, I like that.
The AI isn't painting pixels. It is calculating this invisible map of continuous flowing forces. It looks at the healthy baseline brain and applies these fluid currents of pressure that gradually warp and shrink and stretch the original brain into its future diseased shape.
That's a great analogy. The technical term they use for this is deformation-based morphometry, or DBM.
DBM.
By modeling a diffusion process over a sequence of these 3D deformation fields, the researchers guarantee what is mathematically known as a diffeomorphic transformation.
Diffeomorphic transformation. What does that actually guarantee for the doctor looking at the final image?
It guarantees that the transformation is perfectly smooth and invertible. In practical terms, it means there's a strict one-to-one correspondence for every single microscopic point in the brain across the entire timeline.
Oh, wow.
So the mathematical point that represents a specific ridge on your hippocampus at age 60
Is tracked.
track, locked in, and smoothly mapped to its exact new location at age 65. Because it relies on those continuous forces, like your ocean current analogy, you cannot have tearing.
No more ripping like wet paper.
Exactly. You cannot have impossible overlapping folds. The anatomical topology is completely preserved.
So no more pixelated Frankenbrains. Just
Yeah.
smooth, biologically plausible atrophy.
Precisely.
But wait, I want to challenge the computational reality of this because a single 3D high-resolution brain scan contains a massive amount of data.
Oh, gigabytes upon gigabytes.
Processing that entire volume and doing it simultaneously across multiple time steps to create a 4D sequence, I mean, that requires an absurd amount of computer memory.
It does.
Most standard GPUs would just melt. So how does the AI actually handle the sheer size of a 4D velocity field?
Well, the computational load is exactly why older methods compromised in the past. Previous researchers would either just look at 2D slices of the brain one at a time.
Which loses the 3D context.
Right. Or they would only generate a single 3D follow-up scan, completely ignoring the continuous timeline.
So how did this team fix it?
To achieve true 4D generation, they introduced a frame-wise volumetric patch embedding strategy.
Frame-wise volumetric patch embedding. So they are basically cutting the brain into pieces to make the file size manageable.
Basically, yeah. They take the full 3D volume of the brain and divide it up into tiny 3D cubes, or patches.
Okay.
The neural network tokenizes and processes each of these 3D volumes independently, which drastically reduces the memory burden.
But hold on. If you cut the brain into isolated cubes, how do the edges not look totally disjointed when you put them back together?
What do you mean?
Like if I take a picture of a flowing river, cut it into squares, and edit the flow of the water in each square independently, the currents aren't going to line up anymore, right? The edges are going to clash.
That is a very real problem. And it's the exact problem they solved using advanced spatial and temporal attention mechanisms.
Attention mechanisms.
Think of attention mechanisms as the AI's ability to keep its eye on the ball across both time and space.
Yep.
While the AI is calculating the fluid dynamics for one specific cube of, say, the frontal lobe, the spatial attention mechanism forces it to communicate with the neighboring cubes in real time.
Ah, so it's talking to the cubes next door.
Exactly, ensuring the currents match up perfectly at the borders.
That handles the space and the temporal attention.
The temporal attention mathematically chains the time steps together. So as the AI looks at a patch in year 1, the temporal attention forces it to reference that exact same patch in year 2 and year 3.
Ensuring the disease progression flows logically from one moment to the next.
Exactly. It learns the continuous evolution of the brain structures without ever losing the high-resolution detail.
That is an incredibly elegant way to cheat the hardware limitations while maintaining the physics.
It really is.
But, you know, elegant math on a whiteboard doesn't mean much until you validate it on real human data.
Of course.
I want to talk about how they proved this actually works in the real world.
So the researchers validated the model using 2 massive, highly respected longitudinal neuroimage databases.
The Alzheimer's Disease Neuroimaging Initiative, known as ADNI, right?
Yes, ADNI, and also the OASIS dataset. They didn't just want to see if the math checked out. They wanted to see if the generated brains actually mirrored the clinical reality of Alzheimer's.
I was looking at the visual comparisons they generated in the study, and seeing the timeline of a 62-year-old patient projected out to age 70 is just wild.
That's sobering.
They use these intense color maps to highlight the changes. What exactly are those color maps showing us about the disease trajectory?
Those color maps highlight morphological differences, heavily focused on the accelerated atrophy characteristic of Alzheimer's.
Okay.
When you look at the sequence leading up to 70.5 years, you immediately see prominent ventricular enlargement.
Meaning the ventricles are getting bigger.
Right. The fluid-filled spaces in the center of the brain are massively expanding.
Because the gray matter on the outer surface of the brain and the white matter tracks inside are aggressively shrinking away.
Exactly.
The fluid has to go somewhere to maintain the pressure, so it fills the gaps.
Furthermore, they mapped out the Jacobian determinant to mathematically prove the biology was accurate.
The Jacobian determinant, let's simplify that for the student listener. What exactly is that measuring?
Think of the Jacobian determinant as a highly precise heat map of inflation and deflation.
Okay, inflation and deflation.
A value below 1 indicates that a specific 3D pixel is deflating. The tissue there is actively dying and shrinking.
And a value above 1.
A value above 1 means the area is inflating or expanding, which is exactly what we see happening in the ventricles as they fill with more cerebrospinal fluid.
So the AI isn't just making the brain look older aesthetically.
No, the underlying heat map perfectly tracks the volume changes we expect to see in a progressive case of Alzheimer's.
And the trajectory changes depending on the patient's baseline health, right?
Yes, the model is conditioned on clinical variables. If the baseline scan comes from someone diagnosed as cognitively normal, the generated decade-long timeline remains largely stable.
Just normal aging?
Normal aging, but no aggressive decay. If the patient has mild cognitive impairment, you see gradual structural shifts. And if the baseline indicates Alzheimer's, the AI simulates that aggressive characteristic breakdown.
Okay, so the AI can make a mathematically perfect, biologically sound movie of future brain decay. But how does a researcher actually use these artificially generated sequences?
What do you mean?
Because the ultimate goal isn't just to make a cool video for a medical presentation.
Right, no. The ultimate goal is data augmentation. We got to circle back to the very first problem we discussed, the missing time problem.
The ripped up flip book.
Right. We only have sparse, random snapshots of real patients because longitudinal studies are so hard to run.
And patients drop out.
Exactly. This 4D model mathematically fills in all those missing pages. It creates a continuous, unbroken timeline.
Okay.
And by generating these missing scans, the researchers use this AI to train other diagnostic AI models. They call these downstream tasks.
I love this concept. It's basically AI eating AI.
In a way, yes.
The generative model makes synthetic data to teach a diagnostic model how to be a better doctor. How did they set that up?
They took these synthetic, artificially generated brain trajectories and fed them into a classification model called VGG3D.
Okay. And what does VGG3D do?
The entire job of VGG3D is to look at a brain scan and predict if the patient has Alzheimer's.
Got it.
They also fed the synthetic data into a segmentation model called UNet.
Right, segmentation, which is critical in clinical workflows. For anyone unfamiliar, segmentation is when the AI automatically draws the exact borders around different brain structures, like outlining the hippocampus so the doctor doesn't have to do it by hand.
Right. And by supplementing the sparse, real-world clinical archives with these highly accurate, synthetically generated continuous trajectories, the performance of both downstream models improved significantly.
Because suddenly, the diagnostic AI isn't trying to learn how a disease works by looking at isolated random snapshots.
It has 10 times the amount of data.
And it's watching continuous, smooth movies of the disease progressing.
Exactly. It learns the subtle intermediate transitions between health and disease. VGG3D got better at distinguishing between cognitively normal patients, those with mild cognitive impairment, and those with confirmed Alzheimer's.
And UNet.
UNet got significantly more accurate at mapping the precise borders of shrinking brain structures. The synthetic data made the diagnostic tools fundamentally smarter.
That is just incredible. Pulling back to look at the macro view of this research, the takeaway here for students is that this is a massive paradigm shift in how we approach medical imaging.
A huge shift, yes.
By abandoning 2D algorithms that just smudge pixels around and embracing a 4D model that uses geometric deformation based on actual fluid dynamics and biology, this team has functionally solved the sparse data problem.
They really have.
They can take one single scan from a patient and accurately model a decade of structural brain changes.
It is a monumental step toward predictive modeling.
Right.
Because if you are designing a clinical trial for an early-stage Alzheimer's intervention, you no longer have to guess how a patient's brain might degrade over the next 5 years.
You can actually model it.
You know exactly what the disease trajectory looks like, which means you know exactly what baseline you are trying to alter with your therapeutics.
We finally have the continuous timeline, but, as we wrap up, this technology leaves us with a pretty intense real-world implication to think about.
It really does. The paper proves the mathematics are sound. We can computationally simulate a disease trajectory over 10 years from a single baseline scan. Right now, this framework is specifically aimed at clinical trials and high-risk populations. But advanced medical technology always has a way of trickling down into standardized care.
That's true.
So what happens when this predictive modeling becomes a standard part of a routine clinical workflow?
You mean like scanning everyone?
Imagine you are a completely healthy 30-year-old. You go to the hospital for a routine MRI. Maybe you, bumped your head playing sports, or you get it as part of an advanced physical.
Okay.
And as part of your standard digital chart, the AI hands you a hyper-realistic, mathematically accurate 4D video of exactly how your specific individual brain will shrink, fold, and age until you are 80 years old.
Wow. You would be looking at a crystal ball of your own biology.
You are confronted with the undeniable visual reality of your own inevitable cognitive decay. It is no longer an abstract concept of getting older. It is a bespoke video of your brain atrophying.
That is heavy.
How would seeing your own anatomically consistent cognitive decline change the way you live your life today?
We always want medical diagnostics to be simple. We want a clear picture of what's broken right now so we can fix it. But when the imaging becomes a predictive timeline, the question isn't just whether the math works. The question is whether we are psychologically ready to look at the results.
Exactly.
Definitely something to chew on while you're studying your deformation-based morphometry. Keep asking questions, and we'll catch you next time on The Deep Dive.